Good morning. You can see the screen. Can you please message in the comment box if you can see the screen, if you can hear the voice? See the screen. Can you please message in the comment box if you can see the screen, if you can hear the voice? Oh, thank you so much. Thank you so much. So, a very good morning to all of you. Right, today we'll be discussing about some what you call as MCQs, right, which will help you for your preparation for your entrance examinations, which you are preparing for, right, for your admissions to MSc Microbiology, right. As you know that the entire syllabus for this entrance examination includes everything which you have studied from your first year to your TY, right? So I know that has been a bit difficult journey for you all. Just kidding. I know that you paid full attention while you were preparing for your BSc, right? So I'll not be teaching you concepts because you know that the number of days which are left with us, they are very less, right? So in 2-3 days only, not much, but for around 15-20 minutes I'll be engaging you, maybe in the early mornings or in the late nights. Maybe the time which you don't prefer for studying actually. But then still, let's have an overview of what you have studied in your lower standards from your F5 to TY microbiology, and let us have a brief summary of some things. Like here I can see I have a lot of enzymology, I suppose. Maybe enzymology is my favorite subject, maybe because of that, right? So talking about the first question here, I hope you can read it the screen, and if you cannot read the screen, let me read it out for you. I have only this one mobile with me, right? So enzymes catalyzing glucose 6-phosphate to fructose 6-phosphate, right, will have a EC number. You know that enzyme commission has given numbers, right, to your different enzymes. You have your mobile numbers, but these are different numbers. So enzyme catalyzing the reaction glucose 6-phosphate to fructose 6-phosphate will have a EC number with the first digit of, I'm glad they asked only first digit. If you're preparing for net and set and for some examinations, maybe they'll ask your second, third also. But those which are catalyzing this reaction, can you tell that it belongs to which EC number? The 3, the 2, the 4, or 1 or 5? Anything between 1 to 6, because we know that there are 6 classes. The 7th one added lately, but then the 6 basic things which you'll find in almost all textbooks apart from the 7th one. So the 6 basic classes, right? Out of these 6, this reaction, you can see glucose 6-phosphate to fructose 6-phosphate, right? Can anyone tell it belongs to which EC number? Probably no. You know that there are 6 classes of enzyme. To give you a rough brief overview, you know that the enzymes, they are classified into 6 classes. You have oxidoreductases, then you have transferases, then you have hydrolases, then you have lysases, then you have isomerases, and then you have ligases. So these are the 6 classes of your enzyme, right? So this is EC number 1, EC number 2, EC number 3, EC number 4, 5, and 6. So oxidoreductase catalyzes the transfer, right? Oxidoreductase catalyzes what? Transfer of what? Of electron, of hydrogen, catalyzing oxidation and reduction, right? So that oxidation and reduction is catalyzed by this oxidoreductase, right? So if something is getting oxidized or something is getting reduced, right, so that time it is oxidoreductase. If any group is being transferred, then it is your transferase. If there is hydrolysis of a peptide bond or of any what you call as glycosidic bonds, and that is hydrolase. When you add water and you hydrolyze, when you break the bond, form double bonds, but then you don't what you call as use water for that, it is lyase. When you catalyze isomerization, that is conversion of one molecule into another, isomer, that is isomerase. And when you catalyze joining, it is ligase. So these are the six classes. तो अगर हाइड्रोजन जा रहा है, ऑक्सीडेशन रिडक्शन हो रहा है किसी चीज का, इट इज ऑक्सीडोरेडक्टेज. सो फर्स्ट क्लास इज ऑक्सीडोरेडक्टेज. अगर कोई और ग्रुप जा रहा है, अमाइनो ग्रुप जा रहा है, असाइल ग्रुप जा रहा है, तो दैट इज व्हाट इज कॉल्ड एज योर transferase. If it is the bond is being broken by addition of water, then it is hydrolase. If it is being broken without water, it is lyase. Isomerase catalyzes isomerization. Right here you can see glucose 6-phosphate to fructose 6-phosphate. This is a kind of isomerization. Right, anything like your 3-phosphoglyceride getting converted to dihydroxyacetone phosphate. Right, many things, places where you will find that there is isomerization. So here this is isomerization. So isomerization, if you see, it belongs to which class? अभी आप बता सकते हैं कम से कम कौन से क्लास को belong करता है isomerase? This is isomerase, right? Now can you please comment in the comment box कहाँ पे है ये isomerase? Yes, can you tell where isomerase? Anyone? Isomerase is your class 5. So isomerization, remember oxidoreductase, transferase, hydrolase, lyase, isomerase and ligase. तो हम वहां पे किसकी बात कर रहे हैं? आपकी isomerization. So isomerization is by your fifth number of your enzyme, that is what you call as the fifth class. Identify the pair with the correct group transferred. Right. So which of these is actually transferring a correct group? Like they have talked about CoA. CoA, coenzyme A is, they are saying, is transferring hydrogen. Then NAD is transferring acetyl according to them. Then you have biotin, which is transferring CO2. Then you have thiamine, that is transferring amino group. Right. So you have CoA transferring hydrogen, coenzyme A. Coenzyme A, it doesn't transfer hydrogen, right? Coenzyme A, it transfers your acyl groups, right? You must have heard about acetyl CoA or something like that, right? Then you have NAD, that is nicotinamide adenine dinucleotide, right? It transfers hydride ion. It transfers which ion? Hydride ion, right? Then you have thiamine, right, which transfers one carbon group. So यहाँ पे आपको याद रखना है, अगर आप भूल गए तो I am very doubtful. But then coenzyme A, it transfers what? Acyl groups, right? NAD, it transfers what? Hydride. Hydride, can anyone tell hydride is what? Like it is H plus or it is H or it is H minus? Anyone knows what is a hydride ion? It is H or it is H plus or it is H minus? What is hydride? Yes, very good. It is H minus, right? So hydride is H minus. Thank you so much, right? So NAD, it transfers that H minus, right? Biotin, it transfers CO2. Yes, you are right. So biotin, where you see your carboxylases, they will have maybe this biotin, like acetyl CoA carboxylase, which catalyzes the conversion of your what you call as your acetyl CoA, then to your malonyl CoA you form in between during formation of fatty acids, right? So the only right pair here is your biotin carrying your CO2. So it is biotin which carries what is called as your CO2. So just remember, coenzyme A carries acyl groups, NAD it carries hydride, and thiamine it carries amino group. No, thiamine it carries one carbon groups. Yeah, na? Amino group is carried by pyridoxine, right? Can anyone say that pyridoxine is which vitamin? Vitamin B2, vitamin B3, vitamin B6 or vitamin C? pyridoxine कौन है? B1 है, B2 है, B3 या B6 है या C है? pyridoxine. Wonderful. So pyridoxine is your vitamin B6. So pyridoxine in the form of pyridoxyl phosphate, in form of pyridoxamine phosphate, it is involved in transfer of your amino groups. Anyways, ribozymes, right? Ribozymes, they are what? Right, you must have heard about these ribozymes. You know that most of our enzymes, they are proteins, but now talking about ribozymes. So ribozymes, they are enzymes that cleave your ribose. It may be ribase. So enzymes that cleave ribose, no, it's not. Enzymes that cleave ribosomes, why the hell you will cleave ribosomes? RNA with catalytic activity, yeah, ये थोड़ा सा कुछ लग रहा है. RNA with catalytic activity and ribosomes having nutritional activity. You will eat up all ribosomes. So they are not that. So RNA molecules, remember, which are having catalytic activity, they are called as your ribozymes, right? So most of our enzymes, I told, they are proteins, but then not all of them. We have discovered many RNA molecules which have this enzymatic activity, right? So those RNA molecules which have enzymatic activity, they are referred to as ribozymes. Remember, maybe in protein synthesis, you will find that the larger subunit of your ribosome, which catalyzes the peptidyl transferase kind of activity, has a lot of RNA which is functioning there. Your spliceosomes, which are actually modifying your RNA there, right, removing your introns and joining your exons, right, that is also ribozymes. They are giving an insight that probably the first genetic material which would have come on this planet would have been, what is the probability, like what is the theory which says that what would be the first genetic material which came on this planet? पहला जो genetic material इस planet पे आया था, लोग क्या सोचते हैं आज कौन था वो? RNA, DNA, proteins या फिर carbohydrates? Yes, which was the first genetic material which people think is the first genetic material? Great, right? So that was your RNA. So many people believe that RNA is the first genetic material which came on this planet because it was having both. It was having the ability to replicate, it was having the ability for catalysis. तो दोनों जो abilities थे वो किस में थे? आपके RNA. इसलिए लोग सोचते maybe ribozyme का discovery was one more addition in that particular theory where people started believing that yes, RNA would have been the first genetic material. So remember, ribozymes are RNA molecules with catalytic activity. Next it goes in equation. The enzyme mechanism, like what substrate concentration is needed relative to Km to reach 0.5 of your Vmax. Now, if you want to reach 0.5 of your Vmax, right, what would be the substrate concentration which would be needed to reach 0.5 of your Vmax? अभी जैसे मैंने आपको बोला, if you are talking about say V0 is equal to Vmax into S upon Km plus S, right? This is what we generally have an equation of, right? And actually this doesn't need any calculation because as per the definition, right, हम क्या बोलते हैं that Km, Km is the substrate concentration required to reach half of your Vmax, right? तो हम क्या बोलते हैं कि जब substrate concentration is equal to Km, that means you have reached half of your Vmax. यहाँ पे exactly उल्टा पूछा है, what is the substrate concentration required to reach 0.5 Vmax? 0.5 Vmax is half of your Vmax, right? So when 0.5 Vmax is half of your Vmax, अकॉर्डिंग टू द डेफिनेशन ओनली, है ना. यहाँ पे अगर आप ग्राफ भी देखते हैं माइकलिस मेंटेन का, तो यू हैव दैट हाइपरबोलिक, यहाँ पे क्या होता है आपका Vmax, और यहाँ पे हाफ ऑफ Vmax आप प्लॉट करते हैं, और हाफ ऑफ Vmax पे यहाँ पे जितना सबस्ट्रेट कॉन्सेंट्रेशन आता है आप उसको क्या बोलते हैं, Km. सो 0.5 Vmax, हाफ ऑफ Vmax इज 0.5 Vmax, तो 0.5 Vmax पे आपका सबस्ट्रेट कॉन्सेंट्रेशन कितना होता है, Km. 0.5 Vmax पे आपका सबस्ट्रेट कॉन्सेंट्रेशन कितना होता है, Km. तो डेफिनेशन में ही हो, आपको कैलकुलेशन की जरूरत ही नहीं, सिर्फ आपको अंडरस्टैंड करना पड़ेगा कि इन mm इक्वेशन व्हाट सबस्ट्रेट कॉन्सेंट्रेशन इज नीडेड रिलेटिव टू Km to reach 0.5 Vmax. So Km is the substrate concentration required to reach half of the Vmax. तो यहाँ पे आपका आंसर क्या आएगा, Km. ग्रेट. अब थोड़ा कैलकुलेशन की तरफ जाते हैं. Like here you have what? At what S is V 25% of Vmax? मुझे बताओ, when I'm talking about 25% of Vmax, it is 1/2 of Vmax, 1/10 of Vmax, 1/4 of Vmax, और 1/5 of Vmax. 25% म्हणजे किती? 1/4, 1/5, 1/10 का 1/2. 25% यानी कितना? Can you tell 25% is how much? 1/4, 1/5, 1/10, 1/2. एवढं तर मॅथमॅटिक्स आलं पाहिजे. Quickly. Also, I request you comment your name so I could come to know that who is actually attending the class. So comment your name and comment the place from where you are attending. And I would like to know who is this Ross Anthony. नहीं तो क्लास किसके लिए ले रहे कुछ समझ में आ रहा नहीं. Yes, Vivek, great. So it is 1/4. So when I'm talking about 25%, it is 1/4. तो 1/4. अब उन्होंने क्या बोला, अभी आप इक्वेशन अगर देखते हैं, इक्वेशन क्या है, V0 is equal to Vmax into S upon Km plus S, right? So V0 is Vmax into S upon Km plus S. अब इन्होंने क्या बोला है, what is the substrate concentration for V to 25% of your Vmax. मतलब आपको ऐसा करना है कि V0 is 1/4 आपने बोला ना of Vmax. So now what I'll do is, instead of this V0 I write here Vmax by 4, right? So Vmax by 4 will be Vmax into S upon Km plus S, right? According to Michaelis-Menten equation. Now this Vmax will get cancelled. I'll have 4 Km plus 4 times of S. No, I have what for cross multiplication. If I do, यहाँ पे अगर मैं cross multiply करता हूँ, ये इधर जाएगा. So this will be Km plus S is equal to 4S. So this Km will be equal to 4S minus S. So 3S will be equal to Km. So S will be equal to, comment करो please. S will be equal to now 3/4, 4, 1/3 का 1/4. Km तो है ही यहाँ पे. S will be equal to how much? 3/4, 4, 1/3 and 1/4. I again request you to please comment your names and the place so I could know that whom I am teaching, and also comment, I want to know who is this Ross Anthony, the intelligent guy out there. Yes. So it is 1/3. Very good. Great. So it is 1/3. So at what S is V 25% Vmax? तो आपने क्या किया, 1/4 लिया, ये अपने कैलकुलेशन किया, देख लो एक बार वापस से, इसको क्रॉस मल्टीप्लाई किया तो S कितना आया आपका, 1/3 of your Km. So S is 1/3 of your Km, right? Great. So it is 1/3 Km. Next one. Which inhibitor changes Vmax? Changes Km, sorry. Which inhibitor changes Km but not Vmax? Km चेंज कर रहा है वो इनहिबिटर, पर क्या चेंज नहीं कर रहा है Vmax? आपने तीन टाइप के इनहिबिटर पढ़े हैं, राइट? यू हैव कॉम्पिटिटिव इनहिबिटर, you have uncompetitive inhibitor and you have non-competitive inhibitor, right? अब ये तीन इनहिबिटर में अगर हम देखेंगे, है ना, you have these three and there are two values, right? One is Km, another is Vmax. So in presence of competitive inhibitor, we know that the Km value increases, Vmax does not change. In uncompetitive, both the values they decrease, and in non-competitive this does not change and this decrease. Wow, इतना simple सा आप टेबल भी अगर याद रखते तो I think this answer is now with you. Which inhibitor changes Km but not Vmax? अब इसमें से देख आप सीरियसली बता सकते हैं कि कौन सा इनहिबिटर Km बदलता है पर Vmax नहीं बदलता? प्लीज टेल मी. Okay, intelligent girl. So I would like to know the name of this Ross Anthony, and I would like to also know the other people out there. Yes, tell me which inhibitor changes Km and not Vmax. ये table याद कर लो आप. Remember this table, right? Wonderful, wonderful. It is your competitive inhibitor. Competitive inhibitor, it changes your Km but not your Vmax, right? You can scroll the channel and you will also find a detailed description of these inhibitors. You will find a detailed description of this Km Vmax also. So here it is your competitive, what you call as, yeah, inhibitor, which changes Km but not the Vmax. The next is so simple. In non-competitive inhibition, non-competitive inhibition आपको बताना है Km increase, Vmax increase, Km decrease, Vmax decrease. इसमें से देख के आप सीधा बता सकते हैं नॉन कॉम्पिटिटिव में क्या हो रहा है. Anyone out there? Non-competitive, what will happen? Yes, Prathamesh, competitive. Yes, Nidhi, competitive, right? I would like to go for the next one. In non-competitive में क्या हो रहा है? Km increase, Vmax increase, Km decrease, Vmax decrease. Non-competitive में आपके सामने है. Wonderful. So in non-competitive inhibitor, the Vmax it decreases. So you have option D, that your Vmax it decreases. Great, Prathamesh. The Vmax it decreases. Okay. Let's go for the next one. I hope you remember this now. I'm going out there from this now. So next one which you have is your, if an amino acid weighs 150 Daltons, right? Let us assume, I know it doesn't weigh 150. We generally take a average of 110 or 120, something like that. But maybe ye khate pite ghar ka amino acid hoga. So like, but we have to solve problems according to their assumptions, according to not our assumptions. Anyways, nothing goes according to our assumptions in life. So if an amino acid weighs 150 Daltons, है ना, एक अमाइनो एसिड है उसका वेट कितना है? 150 Daltons. What would be the weight of a tripeptide? Like you have three amino acids together, 1, 2, 3. What would be the weight if one amino acid is 150? What would be the weight of three amino acids together? If one amino acid is weighing 150, what would be the weight of three together? Wonderful. Now I got you there. So 150, like, Anna, so how you calculated it was 150 plus 150 plus 150, but you need to remember that every time the amino acid joins, there is a removal of water molecule. So you have two peptide bonds here. So in these two peptide bonds, two water molecules are gone. So when they will go, they will take the weight with them. So this is 18, this is 18. तो कितना जाएगा? 36. So 450 minus 36. Now can you do it for me? No, 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 no. You can here see 18, 18. पानी को बाहर निकालो है ना. Water is going to go. When a peptide bond is going to form, water will go out. And when water will go out, its weight will also go out. So it is not as simple as 150 plus 150 plus 150. It is 150 plus 150 plus 150 minus 18 minus 18. That is minus 36. What will be the answer? Yeah, so wonderful. Now you got it. It is around 414. So remember, when the amino acids they join, there is a removal of these water molecule, and you get the answer of 416. Great. Oxygen is not involved, not evolved, right? So oxygen is not evolved. We are out of the enzymes, I suppose. So oxygen is not, but not out of biochemistry yet. Oxygen is not evolved during the photosynthesis by, like, who doesn't evolve oxygen? You heard about photosynthesis, that is anoxygenic photosynthesis, the photosynthesis which does not liberate out oxygen. So इसमें से कौन सा photosynthesis oxygen बाहर नहीं देगा? The one which is done by nostoc, that is a blue green algae. The spirogyra, this is a green alga. The rhodospirillum, doubtful लग रहा है थोड़ा. And then you have Finaria. You know Finaria? Finaria is a moss. It is a plant. So which one you think will not give out oxygen? A, B, C, or D? Which one will not give out oxygen? A, B, C, or D? The blue-green algae, the green algae, the seem to be a anaerobic photosynthetic bacteria, and Finaria, which is a moss. A, B, C, D. Great, Priyanka. It is your C, that is rhodospirillum. By all others, they are going to give out oxygen during their photosynthesis. They are oxygenic photosynthetic organisms, right? If the genetic code was quadruplet, triplet आहे तेच कळा ना. आता quadruplet बघा. So if the genetic code is quadruplet, that means it is like AUGA. So four consecutive nucleotides coding one amino acid. So if the genetic code was quadruplet, the possible codons for 20 amino acids would be. So we know if it was singlet, it was 4:1 because there are four nucleotides A, T, G, and C. If it was doublet, it was 4:2. If it was triplet, it was 4:3. If it is quadruplet, it is going to be 4:4. So 4:4 gives how much? Quickly. 4:4. No, no, Priyanka. 4:4 करा ना. किती कंटाळा. 4 into 4 into 4 into 4. एवढं करा. वही या झोपा नका काढू. Don't sleep out there. Do it. Yes, great. So now you got it right. It is 256, right? So you have 256 what you call as genetic codes. We are glad that we have only 4:3, that is 64. Out of 64, there are three which are nonsense. You know? So the three nonsense codons which you know is your UAA, UAG. Can you tell me the third one? The three nonsense stop codons: UAA, UAG, and the third one is A. Great. The start codon is AUG. Can you tell me these codes for which amino acid? The AUG one. Great. Can you tell me AUG codes for which amino acid? The glycine, the proline, the methionine, or Alanine. Methionine. Great, great, great. Okay. So a lot of microbiology stuff out there. So I know that you're fed up with microbiology. But you'd be surprised to know that in entrance examinations, there are also questions on aptitude. So I thought one or two aptitude here and there, and then we'll stop. I know that you didn't like this session at all. But anyways, you have 3, 7, 12, 18. What comes next? 3, 7, 12, 18. What comes next? Three, 7, 12, 18. What comes next? You come next or I come next? Wonderful. It is 25. So this is plus 4, this is plus 5, this is plus 6, this is plus 7. So next would be 25. Great. One more out there. You have 4, 9, 16, dash, and 36. Very simple. 4, 9, 16, dash, and 36. Please. Oh yes, it is again 25 because it is 2 square, this is 3 square, this is 4 square, this will be 5 square. So that is 25. Great. So 25 again. Oh, I got it. This hidden intelligent girl doesn't want to tell her name. No one wants to tell their name except Vivek and Prathamesh, Geeta and Priyanka. Every others are not revealing their name. Maybe Nidhi is there, yes. You're not even telling from where you are. Anyways, I'm not going to come to your house for a lunch, or don't worry. So it is 8, then you have 32, then you have 10, then you have 40, a bit tougher one, 48, then you have 14, and then you have 56. Quickly. You thought you took microbiology and you'll forget mathematics. Mathematics is not going to leave you. Yes. Oh, great. Ross, Anthony, like Ross is your name, right? Great, wonderful name. Or Anthony is the name. Anyways, go ahead with this 8, 32, 10, 40 is a little messed up, I suppose. Yes. She says 12. Let me see. 8 plus 2, 10, 10 plus 2, 12, 12 plus 2, 14. Great. And this is 32 plus 8, 40, 40 plus 8, 48, 48 plus 8, 56, right? So this is plus 2 and this is plus 8. So here you have your 12. Great, great, great. So thank you. Thank you so much. I think I have a class at 8. I suppose it's already 7:42. I still have to have my breakfast. Maybe today late in the night. Late in the night means not very late, but somewhere around 8:30 or 9 maybe. If possible, we can join. Or again tomorrow morning at 7. Like how many of you can join in the evening also? Like we can discuss more MCQs then maybe. How many of you can join in the evening? Can you join in the evening? Like when the day ends, we can learn, and when the day starts, we can learn. Yes? No one can join in the evening. They want to sleep. They want to have a sound sleep and have some good dreams. Anyways, then we can join in the morning also. Ratri join ho shakta. Is it possible to join at night? Yes. Thank you, Nidhi. Thank you, Vivek. Yes, I think Nidhi and Vivek can join in the evening. Okay, evening also sounds good. Okay, so let's meet in the evening then. Thank you so much. I'll update you. You can check my status there. I'll be sharing what you call as these things. If you are not having my number, any one of you, most of you are having, but it is here right there. It is 98236833. 17. So you can just ping me so that I keep on updating on my status when I'm coming live, so you don't miss out the session, right? So it's a goodbye from Professor Girish Kukreja, and thank you so much and have a wonderful day ahead. Bye bye. Take care.